Okay, thank you for the introduction, Sean. So my name is Franco Oitia. I'm co-founder and CEO of Caspr Biotech. We're developing a portable molecular diagnostics platform powered by novel CRISPR-Cas enzymes, and right now focused on taking to market our diagnostic solution for COVID-19. Um, so molecular diagnostics today is mostly limited to centralized labs with trained personnel and expensive equipment. And what we at Caspr are doing is shifting precision molecular detection from the centralized lab closer to the field. And by the field, we mean closer to the patient, to the hospital, point of care, the low complexity facility, the clinic, and even the pharmacy. And we do that through CRISPR. So many of you might know about CRISPR, mostly associating it as a gene editing technology. While the core ability that this biotechnology brings is for its specific identification of DNA or RNA sequences with great simplicity and ease of use. And that's kind of what CRISPR has been all about in nature since as in its natural form. And at Caspr, we've discovered a novel CRISPR-Cas12 enzyme from unpublished metagenomic data from extreme environments, which is not only very different from the ones of the foundational institutions, UC Berkeley Broad Institute, but also has a differential activity in terms of its performance in specific conditions, which we leverage for our diagnostics purpose. So this enzyme is basically a platform in which we can detect in various kinds of samples, blood, saliva, swab, plant tissue, even materials, and detect for specific viruses, bacteria, genetic mutations, and even GMO in this accessible and fast and precise way. And this has been the focus of Caspr since the very first day that the company started in 2019, and we were part of IndieBio Batch 8. And we've done validations for up to 22 targets. And obviously when COVID-19 emerged, we quickly designed our RNA guide and our system for the detection of SARS-CoV-2. We did initial validations with contrived samples and published a preprint about that, and then moved into the validations with clinical samples. In terms of the products that we're developing using this technology, using this enzyme, these are two main solutions. One, which is the most immediate, which we're soon to taking it for FDA EUA, and for which we already have kind of our production pipeline in place, is the CRISPR test kit. So this is basically a lyophilization of our CRISPR enzyme, coupled with an isothermal amplification enzyme, pre-configured for SARS-CoV-2 detection, and with a readout enabled in a lateral flow format. So this is of high value for that instance of point-of-care diagnostics of SARS-CoV-2 in point of care in hospitals, basically. And the second solution that we're developing is our Phantom device, which integrates our CRISPR-Cas enzyme in a lyophilized format into paper, into this paper cartridge. And this is coupled with the device, which the device basically provides some heating components. And with this solution, you're able to perform a fully automated sample to result detection for COVID-19. And these are kind of the two initiatives that we're right now working with. In terms of the value proposition of the technology and the product itself for COVID-19 is the possibility of detecting in less than 45 minutes in a direct from sample format using a direct lysis buffer without the need for RNA extraction. The ease of use of being able to detect without having to use PCR equipment or expensive equipment or external reagents, but rather with a readout which is simplified to a lateral flow, and with a sensitivity and specificity which is comparable to what is currently being performed in labs with qPCR. And these are our most recent results from clinical validation that we've done with the Health Ministry of Argentina, working out with 36 samples, RNA, extracted RNA from those samples, in which we had 100% sensitivity and specificity, both in the detection on a fluorescence plate reader as well as with lateral flows. And this is the equipment that is needed to perform our Caspr CRISPR kit, which is basically tips, tubes, a micropipette, and a heat block. So it really decreases the burden of needing qPCR equipment or needing external equipment to components which can be bought for less than $300, $400. Right now, we have 4 ongoing pilots, including 2 health ministries, such as the Health Ministry of Canada, Health Canada, as well as the Health Ministry of Argentina. We're in conversations with various governments for the possibility of direct purchasing of our kit, which would be kind of our commercialization strategy at the very beginning. And in terms of our timing to market and the milestones that we've achieved so far, we've done the validations with extracted RNA from patient samples, as I showed you before. We already have a contract manufacturing in place and have set up kind of the supply chain for the key components of our kit, included our CRISPR-Cas enzyme that we manufacture internally at scale. We've recently done the lyophilization and the testing for the stability of our components of our kit with a lyophilization partner. And we're right now in the process of doing our clinical validations with our optimized and finalized kit with our partners, not only testing for the application in extracted RNA, but also rather in the direct from sample format with the lysis buffer, for which we already have some initial results. And we expect to do our FDA EUA by late May or beginning of June. And through our contract manufacturing partner and our supply chain that we've set in place, we would be able to quickly ramp up our production to an estimated output of 2 million monthly tests by August of this year, so very quickly. This is a little bit about the team. We've grown very quickly from being 6-7 members at the end of 2019 to now being a team of 24 people full-time. And we're in the process of raising a seed round to accelerate our time to market, accelerate our capacity to deploy our solution at scale within the second half of this year. So thank you for everything.